Okay, hi everyone. Um, so last Friday, we did not have a live Zoom lecture, but I hope all of you were able to see the announcement in Lima where I provided you the link to last semester's lecture 28. Um, and um, and I've asked you to go ahead and just view that video because it's essentially the same lecture, and you can use that with the PowerPoint slides that I've posted on Lima for you to be able to still experience that lecture. Okay. So of course, normally I prefer to give a live lecture. Last Friday, I was actually giving a different presentation that was scheduled at the same time as our class. So let's continue today talking about our immune system and different examples of diseases involving immunodeficiencies. So one of the real standout examples that we can discuss is AIDS, right? So AIDS is acquired immune deficiency syndrome, and AIDS really first became front and center in the public's consciousness back in the early 1980s. So syndrome, what is meant by syndrome? So syndrome means a set of different medical symptoms that might normally not be necessarily related to each other, but they occur together, right? And collectively, they all sort of describe different possible manifestations of a particular, in this case, set of immunodeficiencies. And so we call it a syndrome. The real standout hallmark of AIDS is greatly reduced CD4 helper T cell counts in patients who are suffering from AIDS. And as a result, the T cell arm of our adaptive immune system is highly compromised, right? Because without enough helper T cells in your immune system, you actually cannot do many of the normal adaptive types of responses nearly as strongly as they need to be, including things like the activation and the selection of B cells to be able to select for IgGs, and the coordination of other immune cells to be able to mobilize appropriate defenses against pathogenic infection. Right? So much of that has to do with the role of having helper T cells around, the CD4 positive helper T cells. So as a result, AIDS is accompanied by severe infections from opportunistic pathogens that normally would not be the source of worry, because normally those types of opportunistic pathogens don't make healthy people sick. One thing that I think you've all come to appreciate over the course of the semester is that we are inundated every day with exposures to all kinds of pathogens, right? And the very fact that we don't get sick every single day, right, is a function of us having a strong, robust immune system to be able to recognize things that we are already familiar with, right, as well as things that maybe we are not familiar with, but we can learn how to have a strong adaptive mobile response against, right? A mobile immune response in response to brand new pathogens that might be something that we previously had not been exposed to. So all patients who die of AIDS, they die from these opportunistic infections. So one of these examples is Kaposi sarcoma, or KS. So it's one of these opportunistic diseases that really made the public aware of AIDS. So healthy people with strong immune systems, rarely get KS. The symptoms of KS are these sort of purplish lesions on the face and neck. Right? These are actually tumors that are growing from lymph vessels, the cells of the lymph vessels. So even though it's called a sarcoma, it's not truly muscle tissue, right? Which is often when you hear sarcoma, that's what you usually think of. Right? So KS was something that normally wasn't a real presence in terms of public health concerns, but in the early 1980s, epidemiologists clinicians began to see more and more and more KS cases. Right? And that was really how the public became aware of AIDS. So AIDS is, of course, caused by viral infection. So this is absolutely an example, right, of pathogenic infection leading to an immunodeficiency that is life-threatening, right? Remember before we said that immunodeficiencies can happen if you inherit certain alleles or genetic variations in important genes that code for any of the proteins that are normally involved in your immune system, right? So if you're sort of missing or lacking the performance of a particular protein or component of your immune system, that would certainly be considered a deficiency. And in that case, it might be you inherited that, right, from your parents. But we also said that immunodeficiencies can happen through the course of a normal life through exposure to pathogens, right? So you can be born with a perfectly fine immune system, and then you're exposed to something that causes a deficiency. So HIV is a prime example of that. So HIV was first identified as the virus that causes AIDS in 1983. There are two main types of HIV that typically discussions about HIV and AIDS focus on. One is HIV-1, and this is the strain that causes most cases of AIDS in most countries. This is the sort of what we think of as being the HIV infection that leads to this onset of chronic illness that eventually leads to AIDS. HIV-2 is a much slower progressing form. It takes much longer to progress to AIDS, and HIV-2 cases, I think historically have been more concentrated in places such as West Africa and Asia. So these viruses, these again represent viruses that have made the jump from one species of organism to another. In this case, we believe that HIV made the jump from primates. Right? Other types of primates that are closely related, right, to us as Homo sapiens, sometime in the 1950s is when they believe this jump happened. And it's very likely that the HIV jumped from chimps and other types of monkeys in West Africa. So we know now that chimpanzees are the likely source of origin of HIV-1, and another type of monkey, the sooty mangabe, is most likely the origin of HIV-2. Right? And so even though they think that this jump happened in the 1950s, it probably happened because you had local people who sort of lived with these monkeys, in the rainforest type of setting, in villages where they maybe hunted these types of animals for food. Right? And then as part of hunting and eating these animals, perhaps were exposed to the infected blood, right, from these primates. Importantly, in these primates, of course, HIV wasn't HIV, right? Because remember we discussed that viruses are very well suited genetically to expect to be in a particular host organism. Right, so that means the genes in the virus, right, their genetic sequences are designed so that the products that are encoded by these viral genes are expecting to have the proteins and other components of the host cell be a very specific shape or identity. Right? So originally in primates like the chimpanzee, this would have been a simian immunodeficiency virus, right? Simian meaning monkey, that actually does not cause widespread illness. So in other words, monkeys who have SIV don't necessarily develop the equivalent of AIDS. And that's why this type of virus probably persisted and continues to persist in these animals, right? They represent a reservoir, a natural repository of different types of viruses, right? That just as long as they stay in that species, that virus is subject, right, to the physiology of that particular type of animal, and the epidemiology, the spread of that virus is contained by the population and how severe that virus is, right? So clearly it doesn't kill off chimpanzees readily. Otherwise, the virus might have actually disappeared. But once the virus got into people, right, it mutated, and it became what we know as HIV today. So HIV and AIDS is, of course, a global pandemic that for the last several decades has been a very pronounced public health problem. This data was taken from our textbook, and it's a bit outdated. You can see this data comes from 2012. So it's not so much that I necessarily expect you to know these numbers, but this gives you some comparisons, right, of the global distribution and epidemiology of HIV and AIDS. So the current estimates are that there's close to 40 million people infected with HIV. Right, we expect something on the order of 2 million new infections per year taking place, and tens of millions of infected individuals are expected to die from AIDS, in the next few years. Look at the distribution of HIV and AIDS across the globe. Right, in North America, for example, again, this is 2012 data, so it's already almost a decade old. At that time, you had 1.3 million total cases. Right? They predicted that there would be about 48,000 new cases in 2012 and about 20,000 deaths from AIDS at that time. Right? But compare those numbers to places such as Sub-Saharan Africa, right, where you have 25 million cases in 2012, 1.6 million predicted new cases in 2012, and 1.2 million deaths, right, that year. And you can see the distribution, right, is all over the place. So places that tend to be more first world, well-developed countries that have access to good modern healthcare and health education, right, so Western and Central Europe, right, tend to fare much better than places like Africa or like places in Latin America, right, in South America. So a lot of this has to do with education. A lot of this has to do with access to good healthcare practices to prevent the spread of the virus. And of course, it has to do with access, right, to good quality modern medication to treat HIV infection. Let's take a closer look at HIV. So HIV is a retrovirus. That means it is a virus that contains in it, its genome is in the form of RNA. And in the case of a retrovirus, the name retrovirus means that RNA has to be reverse transcribed back into DNA form, so that the DNA form can then be integrated into the DNA of infected host cells, for example, our body. Right? So HIV belongs to the lentivirus genus because the course of progression of illness is very slow. Right? HIV leads to AIDS over many years. So it is a chronic disease. So it's similar to other lentiviruses. The host target cell inside of the human body are cells of the immune system. So HIV preferentially infects CD4 helper T cells, macrophages, and also dendritic cells. And over time, in almost all infected individuals, right, eventually the immune system cannot fully eliminate HIV, so that there's always a pool of HIV in the body. Right? And of course, over time, if the actual cells that are preferentially being infected and destroyed by the virus as part of its normal life cycle are these critical immune cells, that's what leads to immunodeficiency over time. So these microscope images on the bottom, I think, are both beautiful and terrifying, right? Because this actually represents an infected cell, perhaps a T cell, that inside it's been hijacked, right? So the RNA genome of the HIV virus has been reverse transcribed into DNA. The DNA has become integrated into the genome of the human cell, so that the human cell is now forced to make all the products of the viral genome, including things like all the proteins that make the virus capsule, right? So here is the new virus particle budding off from the cell membrane of the infected cell. And you can see here, right, as it buds off, the capsule of the cell is actually made from the existing cell membrane of the infected cell, right? So in other words, the infected T cell here is giving up part of its cell membrane, because that part of the cell membrane becomes the capsule of the new virus particle. And you can see the new virus particle has its genomic information inside, and the surface is studded with these different glycoprotein spikes, which the virus uses to gain entry into the next T cell that it's going to infect. So over time, as thousands upon thousands of these virus particles each pinch off a little bit of the cell membrane of this T cell, right, the T cell cannot keep up. So literally, the T cell ends up using up, right, so much of its own cell membrane to make these viruses that the T cell just stops existing and dies. Here, let's take a closer look at the genome of a HIV, and really it's just sort of a typical retrovirus in general. So this is not, our immunology class is not specifically a virology class, so I've on purpose stayed away from getting into many of the workings of how viruses function at the level of their genome. You know, if you're interested in this, of course, we do have a virology course here, which goes into this in much more depth, right? So you can consider taking that if our immunology class has really inspired you to learn more about viruses. But for the sake of our class, you know, don't worry about memorizing this table here, right? But what this emphasizes and what shows you is that the genome of a typical retrovirus is very small compared to the genome of, for example, the human genome that's inside of the cells that the virus is going to infect. It's really just, you know, a few different genes that each encode for very specific components, right, of the virus itself, or components that are part of hijacking the host cell. You can see they've color-coded them here. Yeah, it's a nice cartoon, right? These different color-coded things. You can see that there are what they call these the GAG gene that codes for core proteins of the virus. There's this Pol gene that encodes for enzymes such as the reverse transcriptase that will take care of taking the RNA that's carried around by the virus and reverse transcribing it into DNA, right? So that that can become part of the infected T cell's genome. Envelope genes that encode for things like the glycoprotein spikes on its surface, GP120, GP41, et cetera, et cetera, and et cetera. So these color coding is nice because they put it together in this cartoon on the right. Right? You can't ask for anything more clear than this, right? It's really nice how this cartoon's put together. But you can see here, these are the glycoprotein spikes, right? This GP120 that's encoded for and are part of the surface of the virus. And these GP120 glycoprotein spikes are the right shape to bind to certain proteins that are on the surface of different immune cells in our body, like the T cells, like macrophages, like dendritic cells. Okay. Here you can see a nice microscope image, right, that really matches up nicely with the cartoon. And again, you can see these viral spikes. I think all of you taking this class this semester might have particular interest in this type of understanding of virus structure, because there are many parallels here, of course, with the outer structure of this HIV particle and the outer structure of something like the SARS-CoV-2. Right, the SARS-CoV-2 that also has glycoprotein spikes on its surface. But of course, those are a different shape because the proteins that those preferentially bind to are not the ones that they necessarily encounter on the surfaces of immune cells, but those glycoprotein spikes encounter shapes that are on the surfaces of our respiratory epithelial cells, right? For example, our ACE2 receptors that the SARS-CoV-2 virus preferentially infects those types of cells and tissues. So let's look at the replication cycle. Right, so you can see here's an HIV particle with its glycoprotein spikes on its surface that are made up of these two parts, this GP120, the yellow part, and the GP41. Those are the right shape to bind to these different receptors that are on the surface of this gray cell, and this gray cell represents a T cell. So this is a helper T cell. So the helper T cell has CD4 on its surface, right? So that's this stick over here. And there's another receptor known as CCR5. We have not really talked about CCR5 yet this semester, but hopefully you've already kind of realized, right? There are many types of proteins on the surfaces of immune cells that we don't even get to talk about this semester, right? Since our course is really meant to be a good, strong introduction to different concepts in immunology. So CCR5 is another important co-receptor on the surfaces of T cells as well as other immune cells, and it works together with things like CD4. Right? So the glycoproteins of the virus bind to the CCR5 and the CD4, and then that encourages the membrane of the virus, because remember, the membrane of the virus is just the membrane that was taken from another human cell to start with. So it's the same composition, right? The phospholipid bilayer as the surface of the T cell. So the two can just fuse together almost effortlessly, right? And when that happens and it fuses together, the guts of the virus spill into the T cell. And then here you can see some of those proteins inside the virus go to work. So the reverse transcriptase protein, the blue dot, begins to reverse transcribe the pink RNA, right, making viral cDNA. Right? cDNA means basically copied DNA from the RNA. And that viral cDNA gets into the nucleus and it randomly integrates somewhere in the human genome and becomes part of the human genome. Right? Then the T cell activation, right? So if this T cell now gets activated, it's going to start turning on different gene expression, right? And unfortunately, that includes turning on the expression then of genes that are contained within the HIV particles genome here, right? So once that genome of the virus is integrated into our genome, we call that a provirus, right? The complete genome of the virus now part of the human chromosome here. So proteins start being made, right, from that, and eventually these, well, not proteins, RNAs start being transcribed from the viral DNA, and then these various RNAs get translated to make the different protein components of the virus particle again. And then those get packaged up inside the T cell, right? And then they then get packaged in a budding part of the cell membrane, and then it pinches off as a brand new virus particle, right? Okay. So the progression from HIV infection to full-blown AIDS is very slow because, you know, we evolved to be very good at detecting and fighting against viral infection, right? We've learned about that all semester. You have the interferon response that's very good at cells that are infected, sort of changing themselves to prevent the spread of virus particles, and also signaling other healthy cells to warn them so that they can sort of change and maybe be in a state that is more resistant to HIV infection or to viral infection, right? But because, again, the very cells that are being preferentially infected here by the HIV virus are our immune cells, right? Over time, that takes its toll. Right? So if you look at this course here, upon first infection with HIV, in the subsequent weeks of primary infection, the course of the illness is very similar to other viral infection, right? Because at that point, the person who has a normal, strong load of T cells and other immune cells, you would just be fighting this as though it's a viral infection. And so you get, for example, on the y-axis here, right, this green line is showing you the activity of CD4 helper T cells. So after a couple weeks, right, you get a sort of a peak of CD4 helper cell activity, and then those sort of drop off right? And then during this time, for 2 weeks to maybe 6 weeks, you develop some maybe some flu-like symptoms, right? So the person thinks that they just have the flu. During this time, you experience what's known as seroconversion, because of course, in the background, you're selecting for B cells that have Ig, right, that are the right shape to kind of bind to HIV and hopefully, fight the HIV virus and provide you maybe some sort of lasting memory. So at a certain point, that's when immunoglobulins, IgGs, can be detectable in the blood, right? So we say that at this point, the serum, the sample taken from the patient in the serum, it's now converted. You can actually see that there are anti-HIV antibodies present, right? So at first, right, this elevated IgG is providing you protection, right? So it's really successful in slowing the infection down. So that's why it then takes many years, because it never fully gets rid of the viral load. So you always have some HIV that continue to attack things like the CD4 helper T cells. Right? So as levels of CD4 T cells drop over time, right, gradually, right, the ability of your memory immunoglobulins to successfully work and mobilize and stay, strong, that begins to deplete as well. Because remember, the B cells need things like the helper T cells to tell them what to do next, right? And so you can see as the activity of CD4 helper T cells is depleted, right, after maybe a few years, this is what's known as the asymptomatic phase. So this person by now knows he or she is HIV positive. Hopefully, right, they are receiving some sort of care. But it takes many years now before their level of CD4 T cell activity drops below that critical threshold here, right? And this is when they begin to develop more and more symptoms, right? Because all through this time, the person has every day been exposed to different types of pathogens, right? In our normal environment, just like you, just like me, we're exposed to these things, and they've been successful. Their immune system has been successful in protecting them against these things because they've had enough helper T cells around to still be able to have a strong adaptive immune response, right? But once that threshold is crossed, this phase over here in the white, this is when they begin to develop symptoms, right? Certain things that normally wouldn't make them sick start making them sick, right? And then eventually, once those CD4 helper T cell level drops below the really critical level, then their adaptive immune response no longer can function at all, right? And then this is what's known as AIDS, right? Because it's immunodeficiency that has been acquired, right, through HIV infection from possibly many years before. And once they've reached this point, then any opportunistic infection, right, that normally you would have been able to fight off, right, you will succumb to it. You will die of common everyday types of infections. Right? So what is going on then in the background in your immune system during this time? Right? Because already, hopefully, I got you thinking about, yeah, you have B cells, and the B cells are trying to make immunoglobulins, right? And there's so much time for these immunoglobulins to try to be hypermutated and getting better and better and better, right? What is really happening in the back there? Right? Part of the problem is that HIV is a rapidly mutating virus, right? So that means you're stuck with this problem of, as you're selecting for high quality IgGs to be your memory IgGs, if the virus is never fully cleared and is hanging around, then over time it's mutating, right? So certain pockets or certain viral particles might stumble randomly upon mutations that change a particular epitope on their surface that your high quality, high affinity IgGs, right, were designed to detect, thereby rendering those memory IgGs useless, right? And these mutations, right, also enable HIV to be very difficult to really treat if you just use one particular antiviral drug, right? Because if you treat with an antiviral drug, but the virus is never fully cleared, all you're doing is introducing a selective pressure for new mutations that allow the HIV to avoid these drugs to be preferentially selected, right? And so then the virus that's in the person becomes that new form of the virus that is resistant. So in the first 4 to 8 weeks, if we were to look at things like the load of viruses in the person's plasma compared to things like different antibodies, right? Antibodies against, for example, this line here, the lower blue line, are antibodies against the HIV core proteins. Maybe the upper blue line up here are antibodies against the HIV envelope proteins, like the glycoprotein spikes. Right? So the person's immune system tries to make these antibodies, right, over time, and is able to keep them sustained. So the problem is not the B cells, right? The B cells are still trying to do their job, making better and better antibodies, right? The problem is, right, the virus load, right, keeps going up and down, each bump representing a different wave of mutations in the virus that enable it to kind of escape a little bit from what the person's immune system is doing, right? So over time, even though these antibody levels might be high, right, they might be antibodies that don't actually work very well anymore because the virus has changed and the person's body is constantly in this battle, right? Until after maybe 2 to 12 years, then you enter that symptomatic phase, right, where then the load of virus begins to pick up and up and up as you have things like your helper T cells have diminished, and now you can see towards the end, right, the production of things like antibodies and even the activity of cytotoxic T cells begins to taper off. Again, because without the help of enough helper T cells in this 2 to 3 year symptomatic phase, right, B cells are no longer being adequately selected for and being told what to do if you don't have enough helper cells around to tell them what to do, right? So production of antibodies begins to drop off, and then finally you end with the final terminal phase of AIDS. So before 1997, the vast majority of HIV patients would just progress to AIDS, right? But starting in the mid-90s, they began a new regimen of using many different types of antiviral drugs in cocktails to try to hit the HIV from many different angles, so that even if the HIV was mutating, it'd be harder to imagine that the HIV could mutate in a way to actually get around multiple different drugs all attacking it at the same time. So we call this highly active antiviral therapy, or HAART. And so ever since the mid-90s, right, HAART has been a very successful way of treating HIV through these different types of therapeutic drugs. And so an HIV patient who is taking HAART therapy, right, can live with infection now for upwards of 50 plus years, right? They can never clear it entirely from their body. And importantly, they're always on these drug cocktails, right? But they can almost now indefinitely push off progression to full-blown AIDS by many decades. But again, the virus is never fully eliminated from the body. And so the patients have to live, right, with this constant viral load. And of course, they have to live with many health conditions imposed upon them, restrictions on what they can and cannot do in their daily lives, because these antiviral drugs, right, are doing things like really impacting the normal function of the immune system. So when we talk about opportunistic infections that eventually AIDS patients succumb to, right, this is just a table giving an example of common, relatively everyday types of infections that a healthy person would be fully expected to be able to battle and fend off, right? But that somebody with full-blown AIDS would not be able to. So different types of parasites, right? So parasites in the environment, different types of bacteria, for example, tuberculosis, right? Or salmonella, food poisoning, right? Food poisoning, of course, right, can make you sick, maybe viciously sick for a couple days of throwing up, right, and just really hating life. But you fully expect that you will recover, right, from a bad bout of food poisoning, right? But in a patient who no longer has CD4 helper T cells, you're not going to be able to have a successful adaptive immune response. Different types of fungal infections, for example, yeast infections, right, like Candida, or other types of normal fungi, like even athlete's foot, things like that, right, can actually pose a tremendous threat to an AIDS patient, right? And viruses. So viruses, common everyday viruses, like the members of the herpes simplex virus, right, the things that give you cold sores, for example, right, that to a healthy person might just be a inconvenience, a discomfort, right? So for a few days or a couple of weeks dealing with a cold sore. But if you don't have an adaptive immune system to be able to fight that virus and send it back into hiding in the tissues of your body, right, you will die from it. And importantly, different types of cancer. So these are not infections, right? These are just cancer that's originating from inside the body, that a normal healthy person would not develop, right? So things like Hodgkin's lymphoma, including things like Epstein-Barr virus, positive lymphomas, brain cancers, and Kaposi sarcoma, right? Which I started this lecture giving you an example of as probably one of the first high-profile types of diseases that made the public aware that something was happening in the early 80s as more and more patients were presenting with KS. So that might sound very bleak, right? But our immune systems are remarkable. And remember, our immune systems, especially the way that our adaptive immune system works, is all predicated or based upon the notion that we have lymphocytes, B cells and T cells, that are constantly making new versions of antigen-specific receptors, things like immunoglobulins on our B cells or our T cell receptors on our T cells, right? And that every single day you are making hundreds of millions of new variations of those to come out of your bone marrow or your thymus. And even though, chances are you'll never end up having to use a particular one of those shapes on the B cell or T cell, that's the whole point of how we're able to be ready for something brand new, right? Is our body goes completely overboard and has hundreds of millions of these novel shaped immunoglobulins or T cell receptors ready to hopefully pick up on something by chance. Right? So not surprisingly then, there's going to be a subset of HIV patients who, because of the random nature of making immunoglobulin shapes, right, are able to produce antibodies that are super good at binding and neutralizing the HIV virus particle. So it's approximated that a small minority, about 1 in 500 of HIV patients, do make antibodies that succeed in keeping their levels of HIV very low in their bodies. So we call these elite neutralizers because they make what we call broadly neutralizing antibodies. So you can see here, this is a really cool image, right? Because the gray shape here represents the glycoprotein spike, the GP120 spike on the surface of HIV. This down here would be the actual, what was I going to say? The surface of the virus, right, where the spike sticks out from. These color-coded shapes here are real examples of antibodies that have been detected, produced by some patients who belong to this elite neutralizer group. And you can see these antibodies very successfully bind to the glycoprotein spike in a way that prevent the glycoprotein spike from being able to then bind to the CCR5 or CD4 co-receptors on the surfaces of T cells, right? So if you have this spike covered with high quality antibody, whether it's the purple one, the green one, or the orange one here, right, then your body has a much better job of preventing more of your cells from getting infected. The downside of this is that usually these antibodies are made only after about 2 years of HIV infection, right? Because again, most likely these are not antibodies that the patient started off with, right? These are antibodies that through the process of repeated hypermutation, repeated antigen affinity maturation of their memory B cells arrived at these shapes, right? So the person was already infected, and unfortunately it means they will remain infected for the rest of their lives, right? But at least the levels of virus particle in their bodies are kept very low. Right? So this is the product of repeated secondary adaptive responses leading to memory B cells improving their immunoglobulin Gs, right? But it doesn't result in full elimination. However, now there's quite a bit of interest, clinical interest, in using these types of antibody shapes, right, to maybe prophylactically prevent HIV infection in the first place. Right? So in other words, there is the thought that if you have somebody who believes that he or she has been exposed to HIV, right, that if you could infuse that person maybe the very next day, right, with an IV with loads of these high quality elite neutralizing antibodies, then maybe whatever population of virus particles they have in the body, which would start off very low, right, if this is only like a day after they believe they've been exposed, right, then there's a chance that perhaps these neutralizing antibodies can actually eliminate and prevent that small population of HIV particles from really beginning to grow and take over the body. All right. So here's an interesting story. I think you'll like this quite a bit. I mentioned that in addition to the CD4 co-receptor, that another receptor on the surface of our immune cells that HIV, the glycoprotein spike, can bind to is this CCR5. So CCR5 is another way that the viruses can gain entry into our immune cells. And most of the time, it's sort of a combination of the CCR5 also with the CD4, and that's how the spikes on the virus bind. Right? Interestingly, right, about 1% of people of Caucasian descent, in particular those of European descent, right, are homozygous for a mutation in the CCR5 gene. So because of their ancestry, they carry this allele that causes the CCR5 protein, when it's being coded for by the gene, to prematurely stop being coded for. And so it is a truncation. In other words, at the position in the gene that would encode for the 32nd amino acid in the CCR5 protein, right, there is instead a mutation that puts a stop to the translation. So we call it a CCR5 delta 32, right? It means delta mean missing, right? So starting from amino acid 32, these people, their CCR5 protein, there's no more of it. So as a result, their CCR5 mutant proteins are truncated. They're much shorter and they don't function properly because there's not enough of the protein for it to be able to fold up into the right shape. So this then results in these individuals, about 1%, right, of these of people of Caucasian descent, from being mildly immunocompromised. So that means they've grown up being potentially maybe they're always aware. Why is it that I always seem to get sick, but my friends don't, right? Maybe you even sort of built up a reputation in your own family, something like that, right? Oh, he always gets a cold, right? The rest of us were fine, but he always is the one who gets sick, right? And maybe that's because without a functional CCR5 receptor, right, the ability of your immune system to be 100% strong and active is a bit compromised. Fortunately, the CCR5 receptor is a bit redundant, right? There are other immune receptors that kind of make up for it, right? So the loss of a functional CCR5, right, is not that severe, but it does mean you're probably more prone to getting sick. But the amazing thing about this, right, is because CCR5 is one of the prime receptors used by HIV to gain entrance into the cells, right? That means these individuals are actually resistant to HIV infection. So here's a fascinating case study. In 2008, Timothy Brown, who was an HIV-infected patient, he developed full-blown AIDS and developed leukemia as part of sort of the end-stage terminal illness. In order to save his life, his physicians looked to give him a stem cell transplant. to try to replenish some of the stem cells in his body to sort of amplify the amount of normal cells his bone marrow could try to produce, right? But his physicians were really brilliant, right? Because it occurred to them, if we're going to have to get a donor stem cell from a donor's body, right, to treat Tim Brown's leukemia, what if that donor cell that we use as part of the transplant actually has in it the delta CCR5 mutation, right? So therefore, the new immune cells, like the T cells that are produced from the stem cell, would have this delta 32 mutation in it. So it was a widely ambitious kind of experiment, right? But it worked. So through their list of possible donors, matches, right, they identified a donor who actually carried a homozygous delta 32 mutation in CCR5, and that was the stem cell that got, or stem cells that were transplanted into Tim Brown. And amazingly, not only did that cure his leukemia, but it actually resulted in his HIV being eliminated, right? So he went on to be known as the Berlin patient, before his name was revealed. Right? A few years ago, in 2018, a second patient in London also received the same type of treatment approach and was also cured of his HIV. And so to date, these are the only two patients who in clinical journals or in the medical community are recognized as being the only two people who are ever fully cured of HIV. Sadly, Tim Brown did die. I think he died last year through complications of his leukemia coming back, right? But the amazing thing is, right, that he actually died with his body completely free of HIV infection. So how will humans evolve to beat HIV? So think about this. Why did this mutation, this CCR5 delta 32, if it causes people who have this mutation to be immunocompromised, you would imagine that, yeah, how does something like this actually go on to spread in the human gene pool, right? Because you would think any mutation that somehow weakens something like your immune system, right, wouldn't be selected for, right? In fact, it would be weeded out over many generations. So the fact that the CCR5 delta 32 mutation continues to exist to this day in a large population, right, 1% of people of Caucasian descent, that means there must have been some kind of past evolutionary advantage of having it. Right? And so what was this advantage? This advantage was, just in the same way as we now know that the delta 32 mutation protects against HIV infection, it also protects against things like bubonic plague and smallpox. So back in the history of Europe, Western Europe, when things like the Black Death, bubonic plague, right, killed millions upon millions of people, right, people who had this mutation, right, were more likely to be able to survive. And therefore, this mutation got passed on to their offspring. So I want you to think about this. HIV today provides that similar natural selection process, a selection pressure to favor the CCR5 delta 32 mutation in places like Africa, right? And over time, perhaps that protection will spread among the human species, right? And more people will be born with this delta 32 mutation to protect them against things like HIV. So I think it's a wonderful reminder that human evolution continues to this day, right? We are not the end product. We are not the perfect ideal human being that Mother Nature intended us to be, right? We are just one sort of stop along the way of this continuous process. So who knows? Right? Maybe 100,000 years from now, right, the CCR5 delta 32 mutation will cease being regarded as this exceptional mutation. And maybe in reality, this delta 32 mutation will become known as the wild type form of the protein in the human genome, right? Amazing to think that this is what's going on right now, right? And that we are all part of it. Okay, let's stop there.